There we go. We're recording. All right. So this is the chapter 6 and 7 study guide. Okay. So number 25, that one's that's not going to be on there because I don't want to say it's complicated, but it's kind of it's not very intuitive. So 25 won't be on there. We can move on to 17. Cytokinesis typically begins. That's another one that's not going to be on there because, it depends on the source. One textbook might say one thing, one textbook says the other, and it's never exactly the same. So 17 won't be on there either. 14, which of these events occurs during anaphase? Yes, you will probably get some question like this. And that is correct. The chromatids separate during anaphase. Most likely there'll be one question that combines all those, right? So it'll say, in blank, the chromosomes do this. In blank, the chromosomes do that. In blank, the chromosomes do this. And in blank, the chromosomes do that. So it'll probably be, and then your choices are going to be prophase, anaphase, metaphase, or prophase, metaphase, anaphase, telophase in different orders. But anyway, any questions?
Nope.
Okay, number 28. Which of the following is a characteristic seen in prophase 1 that does not occur in prophase 2? And that is correct. Crossing over occurs. Yeah, and that might be on the exam. 18. What is the difference between a benign and malignant tumor? We can go ahead and skip that. That's not going to be on there. That's a leftover from your old textbook. Which of the following will help prevent cancer? That's also not going to be on there. You can skip ahead. Number 7. Which of the following occurs during interphase? Yeah, chromosome duplication occurs.
I think that's right.
Specifically during the S phase. All right. Nucleosomes are made up of DNA and histone proteins. Yes, and if I have that question, if that question's on there, it'll probably be a little bit less specific. So it'll probably just say DNA and proteins, but we'll see. Okay, number 3, chromosomes of eukaryotic cells are found in the nucleus. That is correct. That's pretty straightforward. Plant cell cytokinesis is different. Basically, just think about how a plant cell is different than an animal cell, and it's all about the cell wall. Well, that's the big thing. So plant cells form a cell plate. Yeah, your 39 is correct. It is B. And I don't know if I'll have that question on there or not. All right, 33. Genetic variation is accomplished by all but one of the following. Yep, independent assortment is part of it. Crossing over is part of it. Events of which both of those happen in meiosis 1, so correct. The events of meiosis 2 is the correct answer because there's no meiosis 2 is basically like mitosis as far as we're concerned. 32. One difference between mitosis and meiosis is you have mitosis produces cells genetically identical to the parent cell. Yep, that is correct. And I could reword that, for example. So it might say the difference between meiosis and mitosis is, right? So I could flop them, not that that would matter. And then so the correct answer could be worded where I say something like, I could swap the order. So it might say meiosis produces cells that are not identical, genetically identical to the parents, while mitosis does, or something like that. All right. Any questions so far?
Nope.
All right. 27. Upon completion of telophase 1 and cytokinesis, we'll skip that one. That's again one of those things that's a little bit more complicated than I want to get into. Which of the following occurs during prophase? Yep. So basically, all the stuff logically that has to happen in the first part, right? The mitotic spindle forms, it attaches to the chromosome, the nuclear envelope disappears, the chromosomes coil up, right? All those things that logically just have to happen in the first stage. So yes, and again, D is the correct answer, but for the exam, I might throw in something else that happens during prophase that I just mentioned, like the nucleus and the coiling of the chromosomes. All right, number 12, the correct sequence of stages of mitosis is prophase, metaphase, anaphase, telophase. That is correct, and that will be a test question. And again, there's going to be another one where you have to guess prophase, metaphase, anaphase, and telophase, but it'll be in a different order. So don't let that fool you. There's one question like the one we just covered right now where it asks you to put them in order, and there's going to be another one that says In blank, the chromosomes do this. In blank, the chromosomes do that. In blank, the chromosomes do this. And in blank, the chromosomes do that. Anyway, number 26. During metaphase 1, homologous chromosomes line up. That is correct, because when you're dealing with the first round of meiosis, it's all about the homologous chromosomes. Those are the things that line up. Those are the things that get separated. And then when you get into meiosis 2, that's when you actually separate the sister chromatids like you do in mitosis. During metaphase, yep, chromosomes line up in the middle of the cell. When I was a student, I always just remember that, because I say, hey, that's when the chromosomes met up in the middle, right? That's metaphase is when the chromosomes met up in the middle of the cell. Excuse me. What chromosomes belong to a normal human male? Yep, so we all have 44 autosomes. Yeah, and a male has an X and a Y. So that is correct. And, the question might say, on the exam, it might say what chromosomes belong to a normal human female, and instead of saying 44 autosomes, I might change it to say 22 pairs of autosomes, right? We're saying the same thing, but a different way. Number 30, for a species with 4 pairs of chromosomes, now you're not going to have to do that. Let's skip that. Okay, which is correct. Yep, there'll be some version of this question, and yes, meiosis results in the formation of 4 haploid daughter cells. And there's so many different ways I could say that, right? I could turn that into the wrong option by saying meiosis results in the formation of 2 haploid daughter cells, in which case that would be wrong. And I could make the one above that, D, I could make that say meiosis forms haploid gametes, in which case that would be the correct answer by changing the word diploid to haploid. So again, when you're studying this, don't just memorize that A is the correct answer. Think about how I could turn the correct answer into an incorrect answer and turn one of the incorrect answers into the correct one. But anyway, number 20, homologous chromosomes, that is correct, can carry genes controlling the same inherited characteristics. That is correct. Number 9, the mitotic cell cycle results in the production of 2 cells, each with the same amount of genetic material and the same. Yep, that is correct. Yeah, anyway. 29, anaphase 2 is essentially the same as mitotic anaphase, except that in anaphase 2, we're not going to deal with that. Now, there's not going to be any questions in which you have to know whether the cell is diploid or haploid during meiosis. Like, you definitely need to know after meiosis that you end up with haploid cells as the result. But to keep things simple, I don't require you to know, at the beginning of meiosis 1 or the end of meiosis 1 and beginning of meiosis 2, I'm not going to ask you whether they're diploid or haploid. So moving on to number 11. Which of the following is a stage of mitosis? Yeah, that question is probably not going to be on there because it's too easy. But yes, the correct answer is telophase. And obviously I could put metaphase, anaphase, prophase, and that would also be the correct answer. Yes, sister chromatids are joined at the centromere. I probably won't have that on the exam. Like I said during the lecture, you need to know that word because I'm going to use that word, but I probably am not going to test you on the definition of it. Anyway, number 35, a karyotype would be unable to determine. Correct. You can't determine eye color by looking at chromosomes, and that's all you can do with a karyotype is look at the chromosomes. You can't see the code. You can't see the A's, the C's, the T's, and the G's, so you don't know what the genes are. Therefore, you cannot determine eye color. Oh yeah, sorry. If you go back to that, another thing, if I were to have that question on the exam, obviously I would change it. So it wouldn't be eye color, but just think about anything that requires a gene. So probably something that we talked about during lab. So chin dimple or freckles or ear attachment, whatever it may be, but probably not eye color. Sorry. And then so the next one, 23, chromosomes that do not determine the sex of an individual are called autosomes. That is correct. I don't know what else to tell you. That's just something you have to memorize. During telophase, all the events of prophase are reversed. That is correct. Telophase is the end, so it's like the opposite. Number 1, a blank is an example of an organism that sometimes reproduce asexually by. Oh, that's definitely not going to be on there. So but you are correct. It was a Komodo dragon. So 37, Which of these describes the type of reproduction that is most? Nah, that won't be there either. I'm not going to ask that question. So 22. What chromosomes belong to a normal human female? Yep, and this kind of goes along with that other question. 44 autosomes and 2 X chromosomes. And again, I could reword that to say 22 pairs of autosomes and 2 X chromosomes, right? It'd be saying the same thing. A cell that completed the cell cycle without undergoing cytokinesis, right? So that means it went through mitosis, but the cell itself didn't split in half. So exactly, it would have 2 nuclei. That is correct. A duplicated chromosome consists of 2 sister chromatids. That's correct. There's not much else to say about that. Excuse me. In the human life cycle, yeah, there'll be some version of this, and the correct answer is meiosis produces haploid sperm and egg cells. And I could change that to be incorrect by saying mitosis produces haploid and sperm, and that would make it incorrect. Or I might say meiosis produces diploid sperm and egg cells, in which case, again, that would be the incorrect answer. And for example, the one under it, C, I might change that one to be the correct answer by saying fertilization produces a diploid zygote, right? In which case, that would be the correct answer. So again, when you're studying these, don't memorize the correct answer. Look at the wrong answers and think about how the wrong answers could be reworded to make them correct. Anyway, number 2, ordinary cell division produces 2 daughter cells that are genetically identical. This type of cell division is important for all of it, except correct, right? So all those other things that are listed are what we use mitosis for, and but we don't use mitosis to produce sperm and eggs because that is meiosis. So that is correct. In meiosis, how does prophase 1 differ from prophase 2? That again, we can. I don't want to say I'll skip it, but I'll remind you for the exam, there's not going to be any questions that require you to know if the cell is diploid or haploid during meiosis. So for this, I would reword that question to say, nah, let's just skip it. Yeah. Anyway, number 4, chromatin consists of, DNA and protein. That is correct. And there's not much else to say about that. And then finally, number 24, sexual reproduction in humans, excuse me, allows a haploid sperm cell to fertilize a haploid egg cell. That is correct. And again, I could make that incorrect by changing the word haploid to diploid. And I could, again, the question above it, I could turn that into a correct answer by saying produces a diploid individual instead of haploid. So again, don't memorize the answer. Think about how I could change the correct answers to be incorrect and vice versa. So any questions on that?
No. Do you want me to submit it?
Oh, sure.
Or does it matter?
Yeah, if you haven't submitted it yet, then go ahead.
Okay, I haven't. Okay, then chapter 7. A lot of them are the same as.
Oh yeah, that's actually that's something I need to fix. Again, this is the first semester where we're doing all this stuff is online, and it's also the first semester we're using the new textbook. So yeah, you can just skip that. You're right, it is the same, and that's a mistake that I just need to fix.
Okay.
Here we go. Now we're getting into genetics. What is the key to recognize the recognition of codominance? And you're right. That is correct. The heterozygotes express the phenotypes of both homozygotes. But remember, for that question probably won't be on there. The way those questions are going to be is I'm going to give you some situation, and you have to tell me if it was codominance, complete dominance, all that. So, for example, they'll say, a purple cat, a red cat, and a blue cat mated, and all the offsprings were red with blue spots. And you'd be like, oh, yeah, that is codominance because it's showing both phenotypes. But again, that's how the questions are going to go for those. Number 2, a mating between a purebred purple flowered pea plant and a purebred white flowered pea plant would produce, correct, a hybrid, because all those other words are just throwing you off. Really, that question could just say, a mating between 2 different purebreds produces what, right? The fact that we're dealing with purple flower pea plants, whatever, that does not matter. And the answer is correct, hybrid. Alleles are described as, yes, excuse me, alternate versions of genes, right? So same gene, different version. And most of the ones we talked about were situations in which there was one gene and two different alleles, usually, right? But, and just remember, there are some exceptions where sometimes it's more than two alleles, like the blood group, for example, the ABO blood types. Anyway.
Does purebred mean that they're homozygous?
Yes.
Okay. Homozygous dominant, or does it matter?
It doesn't matter, yeah, because in that case, like what Mendo was doing was, he would cross a homozygous dominant purebred with a homozygous recessive purebred, and then looked at the F1 and F2 generations.
Okay.
What is the key to the recognition of incomplete dominance? So again, the question's not going to be asked like that. The question would be something like a blue cat and a red cat mated, and all the offspring were purple, and that would be incomplete dominance because, like you said right there, the phenotype of the heterozygotes falls between. It's kind of a mixture of both of the parents. Anyway, assume that having 3 nostrils is inherited as a sex-linked trait on the Y chromosome. A man with 3 nostrils has a daughter who has a son with a man who only has 2 nostrils. What is the probability the 3-nostriled man's grandson has 3 nostrils? Okay, let me think about this. We're saying it's inherited as a sex-linked trait on the Y chromosome. Okay, so only the man can carry it. Women don't have Y chromosomes, right? So we don't even need to worry about him necessarily, because then we're talking about his daughter. So whether he has it or not doesn't matter, because his daughter only has X chromosomes. Let's see, a man who has 3 nostrils has a daughter who has a son with a man who only has 2 nostrils. Okay, good grief. So she's normal. She's XX, right? So try to picture this, and you can write it down if you want, but try to picture the Punnett square. She has an XX. He has an XY, and his Y is normal, right? Because he only has 2 nostrils. Therefore, like you said, there's a 0% chance that they're going to have a kid that's going to have 3 nostrils, because it's a Y chromosome thing, and that guy, he only has 1 Y chromosome, which is normal, and that 1 Y chromosome he has was a normal chromosome. And again, the female is not giving any X or Y chromosome, so yes, that is the correct answer. And there might be a question like that. Obviously, I would change it, so it wouldn't be 3 nostrils. It would be some other ridiculous thing. But anyway, any questions about that?
Nope.
All right. In humans, free earlobes are dominant to attached earlobes, and the presence of freckles is dominant to the absence of freckles. If an individual heterozygous for both of these traits were to mate with an individual with attached earlobes and no freckles, what is the probability? Okay. I'm not going to have a question like that because it's not a math class. But basically what that's asking you is you have to figure out the probability of one thing, then figure out the probability of the other thing, and then multiply them by each other because of the rules of probability. And I might have a question sort of like that, but it wouldn't involve, it would be easier. So we can just skip that one. Okay. An individual with a genotype big A, little, so it's heterozygous for A, heterozygous for B, produces 4 different gametes in equal proportions. This is a demonstration of, yes, law of independent assortment, because those A's and the B's don't have anything to do with each other. So when they go through meiosis, they all line up. It's not like all mom chromosomes on one side and all dad chromosomes on the other. They just do their own thing. So yes, 14 is correct. 31. What is the basis of Mendel's laws? And that is correct. Basically, it's all about the chromosomes and what the chromosomes do. Basically, in meiosis 1. Let me read those other choices. Make sure there's not a better answer. Behavior chromosomes during prophase 1. Oh, I see. Behavior chromosomes during nope, definitely not mitosis. The behavior chromosomes during prophase 1, prophase 2. Nope. Behavior of chromosomes during metaphase 1 and that's a stupid question now that I'm looking at it, because D is sort of correct, but then again, so is A. So if that question is going to be on the exam, it's going to be simplified, and the correct answer would be the behavior of the chromosomes during meiosis 1, right? I'm not even going to get into whether it's metaphase or anaphase or any of that. The fact that it's meiosis 1 That's the correct answer.
Okay.
30. Many human traits, such as our performance on intelligence test or our susceptibility to heart disease, are, and that's correct, influenced by gene and the environment. Now, that question's not going to be on there. What you do need to know is when something is influenced by the gene and the genes and the environment, we call that good grief, not epistasis. I'm so tired. Epigenetics. We call it epigenetics, and you do need to know that. But I don't require you to memorize which things are epigenetic. You just need to know what epigenetic means. Anyway, 29, in humans, the inheritance of blank is best explained as being polygenic. And again, just like the last one, you are correct. It is height, but I don't require you to know that. That's just an example. You need to know what polygenic means, which means it's multiple genes controlling one character. But I'm not going to ask you to memorize that example. So number 35, the recombination frequency. Now we can just skip that. We didn't talk about recombination. So no worries there. An individual with naturally curly hair, an individual with naturally straight hair mate. All their offspring have naturally wavy hair. If an individual with wavy hair mates with an individual with straight hair, what is the probability their child will have curly hair? So I like this question because it's requiring you to figure out their genotype. That's describing the phenotypes, and you have to figure out the genotypes and then do a Punnett square, right? So if they have naturally curly hair, then we're thinking that is going to be homozygous dominant. Let's see, spring have naturally wavy hair, so that could be homozygous recessive. Either way, we have two different homozygotes mating, right? So that means all of their offspring, 100%, are going to have a good grief, a heterozygous, right? No matter what, 100% will have the heterozygous genotype. So that being said, what is the probability their child will have curly hair? Correct. Right. Perfect. 0% chance, because again, the logic here, because you're not going to have that exact question, but the logic is you have a homozygous dominant mating with a homozygous recessive, and again, try to picture that Punnett square in your head. That would end up all four of those options would be heterozygous. So again, yeah, that is correct. Anyway, again, for the exam, I would change it. It probably won't be about hair. And yeah. Anyway, next question, unless you have questions about that.
Nope.
36, linked genes are usually located close together on a chromosome. That is correct. There's not much else to say about that. They're linked because that means, like you said, they're close to each other on the chromosome. So usually they do travel together. So for example, if you have a gene that you got from your mom on chromosome 17, I'm just throwing that out there. It doesn't matter which chromosome. If you got to have a gene on that chromosome that you got from your mom, and there's another gene that's really close to it on that chromosome, then you probably have, again, the mom version of that gene because, it's on the same chromosome. And then again, I'm oversimplifying. But anyway, number 18, what data or test would you seek to determine whether or not a trait is sex-linked? Yeah, pedigree would work. That is correct. And I'm not sure I'm going to have that question on there. Anyway, number 6, according to Mendel's law of segregation. I'm glad we talked. So now remember, they segregate, right? So again, using that chromosome 17 as an example, you've got a version that you got from your mom. You have a version that you got from your dad. And when you specifically, not you in general, when you go through meiosis and you produce eggs, half of those chromosomes are going to be from your mom, half are going to be from your dad. So the correct answer is C. Gametes have only one allele copy for each gene. Number 11, an individual who's homozygous. Correct. They have two copies of the same allele. Whether it's dominant or recessive, it doesn't matter. They have the same. 27, Marfan syndrome is the result of inheriting a single allele. Individuals with Marfan syndrome are tall and long-limbed, have both cardiovascular and eye defects. What type of inheritance? Okay. Basically, what we're getting at here, because I probably won't specifically ask about Marfan syndrome, and you definitely don't need to memorize anything about that. The idea here is we have one gene, right? It said that at the beginning. And you have all these different issues, right? So that is, as you put, pleiotropy. Yes, when you have one gene that results in all these different phenotypes, so to speak, then that is pleiotropy. All right, number 8, Mendel cross purebred purple flowered plants with purebred white flowered plants, and all the resulting offspring produce purple flowers. The offspring are all correct. They're all heterozygous because when you mate two different homozygous, and that's what you end up with. And the allele for purple flower is indeed dominant, right? Because they're all purple. So yes, that's the correct answer. There'll probably be a question like that on the exam, except I'm not going to be talking about Mendel or purple flowered plants. Probably I'll just make up some other kind of nonsense, actually, some hypothetical thing. Excuse me. Number 21, achondroplasia is a form of dwarfism caused by a dominant allele. The homozygous dominant genotype causes death, so individuals who have this condition are all heterozygotes. If a person with achondroplasia, so that means they're heterozygous, mates with a person who does not have achondroplasia, so that means they're homozygous recessive, what percentage of their children would be expected to have achondroplasia? This is a good time to remind you, for the exam, you'll probably want to have some scrap paper so you can do Punnett squares. So in your mind, try to picture this Punnett square. We have a heterozygous person mating with a homozygous recessive person. So let me think. There would be there would be a 2-to-2 genotype ratio. Half of them would be heterozygous. The other half would be homozygous recessive. So yeah, 50%. That is correct. And again, it's really hard to explain without showing you, but try to have some paper, some scrap paper. I'll try to, if I can remember, I'll try to have some too for people who forget. But anyway, 28. What is the key to recognition of his trait? His expression is determined by the effects of polygenic inheritance. And yep, that is correct. The trait varies along a continuum in the population. Another way of saying that is if you were to graph it, you would have a bell curve, right? So that's saying the same thing in a different way. But yeah, that is correct. So, sorry, one other thing to say about that. So remember, most of the traits we looked at only had like 2 phenotypes, right? So you either had chin dimple or you didn't. So if we were to graph that, I'm sure you can picture that in your brain, what that graph would look like. It would be a graph that only has 2 bars. So if you were to graph something like height, imagine you broke it down by centimeters. There would be so many different bars because there's so many different heights. So again, that's why it would be on a bell curve. Anyway, number 19. The blank is most commonly found in nature, and that is correct. It is the wild type. There's no other way to say that. It's just something you need to memorize. A purebred plant that produces yellow seeds across with a purebred plant that produces green seeds. The F1 have yellow seeds. What is the expected phenotypic ratio of seed color of the offspring of an F1 cross? Again, the P color, whatever, those are just, it doesn't matter what the trait is, what the character is. It could be anything for the exam. Basically, what we're saying here is we have the F1 generation is heterozygous, right? Because that's what happens when you mix 2 different purebreds. The hybrid is going to be heterozygous. And if you cross, again, F1 with F1, right? So if you mate a heterozygous with another heterozygous, again, try to picture it in your brain what that Punnett square would look like. You would have 1 that's homozygous dominant. You would have 2 that are heterozygous. You would have 1 that's homozygous recessive. And in this case, it's asking, where'd it go? What is the expected? Okay, so the phenotypic ratio would indeed be 3-to-1 because, like I said, the genotype ratio would be 1 to 2 to 1, but the phenotype ratio would be 3 because the homozygous dominant would have the dominant phenotype, and the 2 heterozygotes would also have the dominant phenotype. Number 38. Yeah. Red-green color blindness is inherited as a sex-linked recessive trait. The gene is found on the X chromosome. Can a man with normal color vision father a daughter who is red-green colorblind? So again, try to think of different Punnett squares. Has an XY, and his X is normal. So think of the different scenarios. If he were to, I don't know, let's say he had a kid with a female who was colorblind, which would mean both of her X chromosomes would have the allele, and do that Punnett square. That means nope. So the answer, sorry, let me explain why. I'm trying to picture the Punnett square. So if that man, even if he has a daughter with somebody, a female who's colorblind, so who has 2 of those recessive alleles on her X chromosome, even then, the daughter is still going to have that one normal X chromosome that she got from her dad. So there's a 0% chance that they would have a kid who has color blindness. So that would be C would be the correct answer. And does that make sense?
Yeah.
Okay. To determine the phenotype of an individual who expresses a dominant trait, you would cross the individual. Oh, yeah. So this is just a test cross. Homozygous recessive. Let's see. Where are we? Individual. Yes. Yeah. You would cross. There's not much else to say about that. That's a test cross. And yes, you would cross them with somebody who's homozygous recessive. And really quickly, before we move on, for you and anybody who's watching this video, I'm going to have a question on the exam, and it's not going to make any sense. And there's going to be a long list of words, and again, none of them are going to make sense. And I need to write this down. And the correct answer is going to be banana. And anybody who gets that correct will get a 10% boost to their grade. So it's extra credit that way. Banana. All right. Sorry. So you got that?
Yep.
All right. Because after we're done, I'm going to post this video, and I'm just going to highly recommend to your classmates that they watch it, and we'll see who watches it and who doesn't. Anyway, in humans, the presence or absence of dimples is a trait controlled by a single gene. What is the genotype of an individual who said, all that? Yeah, you are correct. Answer is B. And that whole question, just a bunch of filler. Really, it could have just asked, what is the genotype of an individual who's heterozygous? Doesn't matter what the trait is. If you're heterozygous, you're going to have a uppercase and a lowercase. 33, what is the best explanation for a big B, little b, big C, little c, cross-producing offspring? Yeah, I'm not going to ask that, but I did mention this in lecture. I said whenever you get something that's not the typical expected phenotypic ratio or even genotypic ratio, it's because it's, like you said, linked genes, but that's probably not going to be on the exam. What name is given to the specific location of a gene on a chromosome? And the answer is locus. That is correct. And the easy way to remember that is because it sort of sounds like location, right? For a purebred, a purebred plant that produces yellow seeds is crossed with a purebred plant that produces green seeds. The seeds of all the offspring are yellow. Why? That, like you said, the yellow allele must be dominant, and that's all there is to say about that. Experience with dog breeding. Now we can skip past that. That won't be on there. You don't. Attached earlobes are recessive to free earlobes. What is the probability of having a child with attached earlobes? When an individual with attached earlobes mates with an individual of heterozygous for free earlobes? Oh goodness. So once again, this is just a Punnett square situation, and it's hard for you just have to try to picture it. So we have a heterozygous, uppercase, lowercase, mating with attached earlobes. Sorry. So that is recessive. So homozygous recessive. So yeah, 50%. Again, it's hard to explain that without showing you. So again, for you and anybody watching the video, I'll remind you, you're going to want to bring some scrap paper and something to draw with so you can do Punnett squares. Number 22, which of the following techniques is used to collect fetal? Nope, we're not going to do that one. You can skip that one. Number 40, experience with dog. Nope, we're going to do dog breeding stuff, so skip 40. Number 34, a dihybrid cross produces 30 recombinant offspring. No, we skip that. I won't be on there. Excuse me. Yeah, 32 might be on there. It's pretty straightforward. Linked genes violate Mendel's principle of independent assortment because they don't sort independently because they're close to each other on the same chromosome. So yeah, that's all you can say about that. 16, a couple has 2 female children. What's the probability of their next? I don't like this question because you could argue it. Yeah, I don't want to waste your time getting into it. So that question won't be on there. Number 12, which of these crosses will produce only heterozygous offspring? And that is correct. The only way you can have 100% heterozygous offspring is if you have a homozygous dominant mating with a homozygous recessive. And when in doubt, if you get this question on the exam and you don't remember, you haven't memorized the answer, just do a Punnett square. Do each one of those, a Punnett square for each one of those, and see which ones have only heterozygous offspring. 24, an individual with naturally curly hair again, and an individual with naturally straight hair mate. All of their offspring have naturally wavy hair. What is the relationship between the alleles for their texture? Yes. Incomplete dominance, because it's kind of a mixture of the two. And that is kind of how the exam will go. Again, I'll just give you some random hypothetical mating and say this is the result. So is it codominance, incomplete dominance, pleiotropy, so on and so forth. Number 7, attached earlobes are recessive to free earlobes. What is the genotypic ratio is expected with an individual with attached earlobes? So that's what homozygous recessive mates with an individual heterozygous. So I think that's the same question from earlier. Anyway, try to picture it in your brain. That Punnett square would be a homozygous recessive mating with a heterozygous. And yes, that is correct. There would be a 1-to-1 ratio, or as we wrote it in lab, a 2-to-2 ratio. So it might be either way on the exam. Remember, those mean the same thing. 1-to-1 is the same as 2-to-2. For the exam, I'd probably write it as 2-to-2 as opposed to 1-to-1, because that's how we did it in the lab, and I want to keep things consistent. Anyway, hypophosphatemia, vitamin D resistance, rickets, is inherited as a sex-linked dominant trait. So that means you only need one of the alleles to have it. You don't even need 2 of them. The relevant gene is found on the X chromosome. What is the expected outcome of a cross between a homozygous recessive woman, so that's 2 normal X chromosomes, and a man who has it? So he has his one X chromosome has it. So again, try to picture the Punnett square in your brain. XY. So 50% chance. Man, I'd have to actually draw that out. Let me think. I don't want to waste your time either. Okay. On the offspring, XY, normal X. Oh man, I need to draw it out. You want to come back to that one?
You can draw it out if you want. I have time.
Okay. Some of these I can picture. Some of them I can't. Let me go grab a pen. Sorry.
Okay, you're good.
All righty. So the female doesn't have it. So that is XX. I'll just use the letter N. So little n. He's X big N, Y. So X big N, X little n. X big N, X little n. X little n, Y. And then X little n, Y. Okay, so I'm looking at this. I'm not even looking at the question anymore. You would have 2 female daughters who have it, because it's, they would be heterozygous, and this is a dominant trait, so that means they would have it. And 2 males who do not have it, because they obviously got their X chromosome from their mom. Anyway, so let me look at these options again.
So it'd be B.
50% of their daughters. No, actually, I think it would be all their daughters and none of their sons. Yep. A would be the correct answer. Which really stinks, because without me showing you the Punnett square, it doesn't really do you any good. But so again, I'll just remind everybody, have something to write with, because you're going to need to do Punnett squares. Not a bunch, but at least a few. The best definition of purebred plant is one that self-fertilizes to produce offspring identical to the parent. That is correct. There's not much else to say about that. An individual heterozygous for cystic fibrosis is a carrier. Oh, that won't be on there because I told you when we talked about cystic fibrosis that was just an example and you didn't need to memorize anything about it. So that's it. Any questions about that chapter?
Nope.
We're moving right along.
I think that's all. That's 6 and 7 and 8. Do we have 9 too?
Yeah, the one that we're doing now is going to be on the exam. Like
Okay.
the chapter we're covering now will be the last chapter on the exam.
Okay. I did it, but this one's harder. Okay.
There we go. All right. The shared genetic code of all life on Earth is evidence that is correct. All life shares a common ancestor. I'm not sure if that question is going to be on there or not. Anyway, number 34, what is the ultimate source of all diversity? And that is correct. It is mutations. And that'll be really important in the next stuff we're getting into after this exam. But anyway, what is the name given to the collection of traits exhibited by an organism? That is correct. That is your phenotype. Number 8, if one strand of DNA double helix has the sequence G, I should wear my glasses. GTC CAT, what is the sequence of the other strand? So remember, A's go with T's and C's go with G's. So that would be a CAG GTA. So yep, you chose the correct one. Obviously, for the exam, I'm going to use different letters, but yeah, and there will be a question like that where you have to do that. I'm not sure if I'm going to ask you any questions about HIV. We haven't covered it in class yet. That'll be hopefully on Wednesday. But the human must use its own reverse. Yes, reverse transcriptase is the correct answer. And actually, I need to look at the new textbook again to see if they even discuss HIV. And if it's not in the new textbook in chapter 17, then it's definitely not going to be on the exam. But anyway. What is the smallest number of nucleotides that must be added or subtracted to change a triplet group? We haven't talked about this yet either, but yes, the number is 1, right? So if you add or subtract a nucleotide, then you're going to change the genetic message, and we'll talk all about that on Wednesday. In a DNA double helix, A pairs with T and G pairs with C. So you are correct. It's T and C. And obviously, again, for the exam, I would change that up to where, there would be a different correct answer. Number 15, how many nucleotides make up a codon? That is correct, 3, right? So all the words in this language are made up of 3-letter words. Whereas translation accomplished, and that is correct. It is in the ribosomes do it. Number 29, during translation, what is the correct order? Codon recognition, peptide bond formation, translocation. That is correct. And yeah, that question might be in there, and it's pretty straightforward. We talked about it today. The backbone of DNA consists of a repeating sugar phosphate pattern. That is correct. That's why it's called the sugar phosphate backbone. Number 32, a mutation within a gene that inserts a premature stop codon. So I talked about this today, actually. In the mRNA would result, like you said, that is correct, a shortened polypeptide chain, because once that stop codon is read, then that's the end of the process. So that's exactly correct. The absence of a terminator. So this is kind of like the opposite. The absence of a terminator in transcription. So we're talking about making an RNA strand here. Results in what? And that is correct. The production of a longer RNA molecule. Yeah, that's correct. How can bacteriophage DNA spread from cell to cell without causing cell death? We're going to cover that on Wednesday. I'm not sure it's going to be on the exam or not, but that is correct. So when viruses attack cells, there's 2 different cycles. There's a lysogenic cycle and there's a lytic cycle. The lytic cycle is shorter because it just results in death right away. And the way I remembered that as a student is the word lytic is shorter than the word lysogenic. So I just remembered lysogenic is the longer one because it doesn't kill the cell right away. Anyway, 38, plant viruses. And I'm not sure I'm going to have this on the exam either. Probably not, but often use RNA rather than DNA. That is correct. Yeah. Anyway, not much else to say about that. 17, transcription is the manufacture of a strand of RNA complementary to a strand of DNA. That is correct. Not much else to say about that either, right? Transcription is how we make RNA from DNA. The region of DNA where RNA synthesis begins is the promoter. That is correct. That is the spot before the gene that the RNA transcriptase, RNA polymerase attaches. Number 43.
We'll get there when.
Oh, okay, yeah,
I'll come back to it.
That's out of order there. Anyway, number 35, mad cow disease is caused by. That's probably not going to be on the exam, but that is correct. It's caused by prions. Number 2, DNA and RNA are polymers composed of correct nucleotide monomers. That's almost definitely going to be on there. Hopefully everybody will get it. That should be an easy question. Number 10, what type of chemical bond joins the bases of the complementary DNA strands? That is correct. It's hydrogen bonds. And that's not trivial, right? Because that's how it allows the DNA to open up, whether it be for DNA replication or transcription. Number 12, DNA replication requires the cooperation of over a dozen enzymes and other proteins. That is correct. Let me look at these other options, though. You require DNA polymerase and RNA polymerase. That is incorrect because you don't need RNA polymerase. It's a very fast process that results in numerous errors. That is incorrect, but I could make that the correct option by saying is a very fast process that results in few errors. In that case, that would be true. And then finally, is a slow process that results in, again, if you were to combine A and C, you would have a correct answer there. So again, for the exam, I might change the words. So don't memorize that answer. Understand what is correct and incorrect and the subject. Anyway, number 31, scientific research companies sells kits that allow researchers to produce proteins in test tubes via a process known as in vitro translation. Which of the following components is not needed? That is correct. If you're just doing translation and you're starting from that step and you're not doing transcription, you do not need DNA, but you do need the ribosomes, you do need the transfer RNA, you do need the amino acids. If adenine makes up 20% of the bases in DNA, double helix, what percent of the bases is guanine? All right, so if there's 20% is A, then that means we also know that 20% is T, right? So we know that 40% is going to be A's and T's. Therefore, it would have to be 60% is C's and G's, right? Because 40 plus 60 equals 100%. So if 60% are C's and G's, and you cut that in half, that means 30% is correct. That it would be G's. And there will be a question like that, but the numbers will be different. Consider the following figure. It illustrates the single amino acid substitution. Well, obviously there's no picture. So
I can find it. That's the picture.
Okay. I see that now. All right.
And then that's a question.
Okay. Consider the following figure. It illustrates the single amino acid substitution in hemoglobin gene. Yes, that is correct. It would alter the protein so it no longer functions properly. I don't like that question. It's probably not going to be on there. As an experiment, you infect bacteria with an unknown virus to see how the bacteriophage affects the bacteria. And the bacteriophage is a virus that attacks bacteria. After a few days, you notice that the bacteria population is growing normally and the cells appear healthy. Assuming that the infection occurred, what can you conclude? And you are correct. Lysogenic cycle, because that's the longer one. It's the one that doesn't result in death. So if I were to reword that question, I might say something like, after a few days, you notice that all the bacteria are dead. And then the correct answer would probably be something about the lytic cycle, because that's the one that kills them. Anyway, number 30. Peptide bonds form between, that's correct, amino acids. They're just covalent bonds. We give them a name when it's between amino acids, and it is a peptide bond. Translation converts the information stored in RNA to polypeptides. That is correct. Which of the following would not likely develop into an emerging virus? That won't be on there. We can skip that. The figure below shows the flow of genetic information in a eukaryotic cell. The transfer of information from DNA to RNA is known as transcription. That is correct. That's just something you have to know, right? DNA to RNA is transcription. RNA to polypeptides is translation. Figure below shows the flow of genetic information in a eukaryotic cell. I already did that one. Sorry. Number 24. The expressed regions of eukaryotic genes are called exons. Yes, that is right. Those are the parts that are expressed. Number 39. A blank is to bacteria as a blank is to an animal cell. Prophase, provirus. That is correct, but that won't be on there. This will not be on the exam. 19, which of the following enzymes is responsible for RNA synthesis? That's straightforward, and that is correct. RNA polymerase makes the RNA polymers. Pick for number 42. Evidence for the spiral nature of DNA comes from, that won't be on there either. Like I said, I wasn't going to ask you, I'm not going to ask you any history questions. 21, the correct sequence of events occurring during transcription is initiation, elongation, termination. That is correct. There's not much else to say about that. Number 14, how many amino acids are common to all living systems? And again, this is just a straightforward one. Yes, the answer is 20. Thymine and cytosine different from adenine and guanine. That won't be on there. When I discussed it, I did tell you that some of them are single ringed, some of them are double ringed, and they even have names for single versus double ring, but you do not need to know that for the exam. So 43. Examine the genetic code table shown below. The codon AGC codes for the amino acid. Oh, yeah. We can come back to that. But basically, you just use the wheel. And yes, there might be a question like that where I give you a code, and then you have to use that wheel to figure out what the code, which amino acid it's coding for. Number 5, RNA contains a nitrogenous base U instead of T. So, yeah, that is correct. The answer is B. Speaking of which, when you're studying, you can just study A, T, C, G, U. You're fine if you just know the letter, because I'm not going to trick you and have like 2 different words that both start with T, right? So you just memorize the letters, and that'll be fine. The RNA that is translated into a polypeptide is, in fact, the messenger RNA. That is correct, the mRNA. The DNA bases AGT codes for an amino acid carried by tRNA with the anticodon. So basically, it's asking what the opposite of AGT would be. So that would be UCA. So wow, it's not even an option. So yeah, looks like that is a misprint. So I need to fix that. Anyway, yeah, there might be a question like that, but the anticodon would be, again, the complementary base, right? So that's why I said, U goes with A, C goes with G, and A goes with T. So that's why I said that. But again, that's not an option for some reason. Anyway, number 11, after replication, each new DNA double helix consists of one old strand and one new strand. That is correct. That's pretty straightforward. There's not much else that you can say about that. If a strand of DNA has a sequence AAG-CTC, transcription will result in, so again, like I've been saying in lecture, anything that has a T in it, you can go ahead and not even look at it because we're making RNA here, so there are no T's in RNA. So let me think. That would be UUCGAG. UUCGAG. Yep. So that is correct. Please read the following scenarios and answer the following questions. No, we're not. No, we won't have any of those questions where you have to read something and then answer questions about it. So we can skip that.
Okay, that's all of them.
All right. Well, we did it. Do you have any questions about anything we covered or the exam? Or?
Nope.
All right. Well, thanks for meeting with me, and thank you for letting me record it. Your classmates will appreciate that.
Thank you.
You're welcome.